Okay, so now that we know what experiment we need to set up, let's just remind ourselves what it is. We ended up deciding that we were going to run our experiment with different time points and different drug treatments. So there were 6 samples that we decided on. We decided to have an untreated control and 2 different types of positive controls. One for making sure that we know what it looks like when PRMTs are inhibited with a general PRMT inhibitor, MS023. And then a second where we know what protein expression will look like when we activate the apoptosis or autophagy pathway by using a proteasome inhibitor, MG132. So those are 2 different ways that we're going to be examining for protein expression that happens in our suspected pathways, and then we'll compare it to our experimental system, which is with our own drug. And our own drug is the P2 compound. And we are going to look at a dose-dependent response, so whether or not there is a dose-dependent response, by running 3 concentrations of our P2 drug, which we are just going to call low, medium, high. In our case, those drug concentrations are going to be 10 micromolar, 20 micromolar, and 40 micromolar. And those are the ones that we have decided on based on previous results from our cell morphology experiments. We may choose to lower this down a little bit to get a broader spectrum or enhance it if needed. But for now, those are the 3 concentrations. So we are looking at a spectrum of concentrations. And so because we have 6 samples, we decided that we were going to use 6 well plates. And we're going to have 1 plate per time points. And our time points are going to be 1 hour, 3 hour, 6 hour, 12 hour, 24 hours, and then the last one is going to be a 48-hour time point. We are only going to treat our cells 1 time with a P2 drug because it is thought that our drug, based on its structure, should be irreversible. So we don't have to do repeated or daily or every 6-hour treatments. And so we'll just do it 1 time and then look at it over time and see how quickly the effect happens and how long it stays, and if there is a dose-dependent effect as well. So that's a general experiment. So that means that we need to be available for several hours, over a couple of days at least, to be able to examine the experimental results, to be able to extract our proteins, to treat the cells, all that good stuff. So now we need to decide, when do you want to start it? Now, depends on people's schedules, right? Whoever is doing it, if it's just one person doing and that's their only job, or are they also taking classes if these are students, or if they have work responsibilities, family responsibilities, all those things need to be taken into account. So typically in a lab, what we will do is anyone who's going to be involved in the project, we will get together and we will look at each other's schedule and we will set up a plan and decide when it is that we're going to be starting our experiment. So we know that we need time. It's going to need about an hour or so to set up our actual experimental plates and take the cultured cells and plate them for the experiment. The setup is fairly easy for this one because as long as we are getting about 40 to 60% confluence on our wells, we are good. The only important part is that all the plates should start off with the same number of cells. So that's a very, very easy setup to do. We don't need to worry about counting cells, calculating exact number of cells to plate, as long as we're plating, based on our experience, 40 to 60% confluence to start with. But that still needs about an hour on the first day. After that, you need at least an overnight time period to allow the cells to attach, kind of stabilize, and start off and set up the cell culture. So we allow overnight for attachment. And then we can pretty much start any time after that for our treatment. For overnight attachment, overnight typically means approximately 16 hours. That's how long you want to wait before you actually treat the cells, to give them enough time to recover from the trypsinization and get settled. Then once we start, then there are 2 different ways that we could do these time points. The first way is that I start my treatments at the same time. So on the treatment day, essentially, in that case, on the treatment day, I'm going to take all my cells and start them all with the treatment. So I will remove the media. You will then wash them with PBS to remove residual serum proteins from them. And you can do it once or two or even 3 times, depending on how much of that serum you want to remove. And then you're going to switch them to treatment media. Now, treatment media is typically the same base media, whatever it is that the cells require, but with a lower concentration of serum. And that typically allows us to enhance the effect of our drug that we are looking at. In our case, we are going to be doing 2.5% serum containing media, or FBS. We don't want to lower it completely. We don't want to make it serum free, because one of the effects is autophagy. And just not having serum can trigger autophagy as well. So we want to keep it where there's a little bit there to keep the things in control, and then we are going to be able to look at the effect of our drug more specifically. If we were interested in proteins that were secreted by the cells, those proteins would have to be collected in the media, in which case we would have to go to serum free. But in this case, we don't need to worry about that. But we'll lower it from about, most people grow their cells in 10% FBS, so you're going from 10% to 2.5%, or 5% to 1%. So you're lowering it significantly to look at the effect. And then this treatment media will also contain whatever treatments you need based on our experimental design. And once you set that up, when you finish setting it up, that's the point that your actual experiment starts. So if you finish setting it up at 9 a.m., then that's your 0 hour. And now you start from there. Exactly 1 hour later, you're going to collect your 10-hour, your 1 hour treatment, then 3 hours, so on and so forth. But that means that that day is going to be a very long day. And you want to make sure that you start early enough that all your hours, up to 12 hours, that you have those 12 hours in a time frame that you will be available. That is not like 2 in the morning, some unholy time that you cannot be there safely, depending on where you live and who you are, and that you will be able to come in and take care of those cells. If you start the treatments all at the same time, that itself is going to take you the longest. So let's say about an hour to 2. But once that's set up, each time point should only take you about 15 minutes to extract and get back in the freezer. It's not going to take you very long. But you do have to be there exactly at that time to be ready to extract. So that's one way to do it. A different way to do it is to treat them in a staggered manner and then collect them all at the same time. The advantage of that is that, remember, we plated our cells at about 40 to 60% confluence. If I treat them all at the same time, my 1 hour, 3 hour, those earlier time points may not have a lot of cells in there. It's not like I will, and my goal is to get as much protein extracted after responding to the treatments as possible. So I may have issues with levels of protein that are present there. And as compared to something at 12 or 24 or 48. By 48 hour, you might be getting the death response from your drug already, so you may already be struggling anyway. But for the other time points, it may be affecting them more than you want. So if you stagger your treatments, the advantage is that all of them get extracted at the same time. So there's less variability from one time point to another because of number of starting cells. There's also less variability from you, because each time you come in, you're in a different mindset. You might be doing something very quickly one time because you're in a hurry to get to class. The next time you may come and you might be tired or relaxed, depending on what it is that you're in mood in. You might be having some variability from that as well. And the third reason is that each time point, once you collect and put it in the freezer, is going to be in the freezer for a different amount of time. Best results are found when you treat everything the same way. So by treating them in a staggered manner, but collecting the media, the extracts of your protein at the same time, you kind of get closest to that story. So at that time then, you would have made sure that they were in the incubator the same amount of time. They were all extracted at the same time, so they went into the freezer at the same time. And so there'll be a lot less variability in your samples. Cool? So let's make an example setup based on our experiment. So we could have started our experiment in the morning or at night. So I'm going to make, 2 columns, one for starting the experiment at 8 a.m. and the other one setting it up. It doesn't matter, for example, well, setup doesn't really matter. So we'll set up at 8 p.m. or in the evening, let's say even 6 p.m., doesn't matter when. At the end of the day, before we are going home, we are setting up our experiment. The next day, let's call it day 0 or treatment day, I have 2 options. I could start it at 8 a.m. That's nice, early morning, right? Because I want to make sure I'm there all day for that. Or I could start it at 8 p.m. Sure. What happens if and so that's the second part. In my case, I'm going to stagger the treatment, and I'm going to start off with the latest time point, okay? It's because I want to collect all my samples at the same time. So the first one that I need to treat is the 48-hour one. And so I'm going to go ahead, I need to, for example, treat the cell. So if I start the treatment here, so this is going to be my 48-hour time point, yeah? I can start it at 8 a.m. or p.m. Oh, let me just put that. Treatment, day 0 treatment, 48-hour time point. So the next one is 24 hours. If I'm doing 8 a.m., then it would be 8 a.m. on day 1. Or let's set up, going to be day 0. This will be day 1. This will be day 2. Yeah? And so it would be 8 a.m. If I was going 8 p.m., then the 24-hour will be 8 p.m. on this next day, right? So you have these 2 days now. Then on day 3 is my long day. So I need to be aware when I am starting. Yeah? And so if I start at 8 a.m., my 12-hour time point, actually let me move day 3 out here because a lot of time points are going to be in day 3. You will have 12-hour, 6-hour, 3-hour, and 1 hour, right? Okay. So if we started at 8 a.m., the 12-hour is going to be 8 p.m. 6-hour is going to be 6 hours from then, so it's going to be what, 2 a.m.? Yeah. Oof. I think we are having a bad night. Next one's going to be 5 a.m. And then 1 hour is going to be 7 a.m. And then after staying up all night, essentially, or having trouble sleep at all night, my extractions are going to be at, and because I'm extracting everything at the same time, that's going to be a couple of hours, 8 to 10 a.m. minimum, maybe 11. So that's a very, very long day at the wrong time. Now let's see if it works better. If I start those first few at night, again, if I'm just treating one plate, it's not going to take long, 10, 15 minutes max. So my 12-hour will be 8 a.m. So the same timings, but now it's a lot calmer. It's in the daytime, right? And even though it goes late into the evening, you have a late day, but it's manageable. It's not too crazy. So you get 5 p.m., 7 p.m., and then 8 to 10 p.m. So the longest, still a long day, but you get a lot more manageable timings than others. So based off of this, and then you would want to look at this and say, okay, clearly we want to go with this pattern. So we want to set them up in the evening and do these first couple of timings in the evening so that we can have a manageable day. And then the last decision to make would be, when do we start? Do we start on a Monday or a Tuesday or a Wednesday? And that's going to be dependent on your group and your people that are working on it and your schedules. So depending on when people can be around, you may choose, maybe somebody has an exam on Wednesday, and that happens to be day 3, and that's not a good day. So we might want to arrange it so that we start on a Tuesday and then have these days on those days that they are busy, so it's quick and easy and simple, and then they can focus on this experiment afterwards, the day after their exam or the couple of days after their exam. So those would be things to consider when deciding when to start your experiment.